So in addition to the small pieces of equipment that you'll be using in the Bio 120 lab, you'll also be using a few larger pieces of equipment that can be found on the back or side bench. Only one or two of these will be in each lab, depending on what lab room you're in. The first piece is a water bath that will be usually set to temp that you're going to use it at for a particular lab. And there will be some water that should be, the water bath should be about half filled with water. And what you're going to do is you never want to throw a tube or a sample just naked into the water bath. What should be hanging out near these water baths are these foam floaties. And what these will do, what you do with these is that the larger Eppendorf tubes along with the smaller Eppendorf tubes will fit into these floaties such that the sample containing side will be down and the top will be kept up and out of the water and dry. And so you want that to do, you want to do it that way so that the top of the tube never comes in contact with the water, because with that you run the risk of some water getting into your sample and contaminating your sample. So you always want to use a floaty of some kind. The other pieces, larger pieces that you'll find on the back or side benches are centrifuges. A lot of times, a lot of protocols call for you to either spin down or pellet something. And so by exposing your samples to a high G-force in these centrifuges that are going to spin your samples very, very quickly, you can actually separate out various things like proteins, DNA, or cells. And so you'll notice that both of these are off right now, and when they're off or unplugged, the top is actually locked shut. And so when you turn these on, the top will then open, and you'll see the rotor inside that has 18 holders in it, and your Eppendorf tubes fit right into those. Your large Eppendorf tubes do. And when you're loading these centrifuges, any of these centrifuges, you want to be sure that you're always balancing them. And by balancing them is that, I mean that you're never going to add just a single tube to one holder in the rotor in these centrifuges. You want to have an equal distribution of weight across the rotor because, as I mentioned, it's going to spin these samples very quickly. And so, kind of like an unbalanced load in a washing machine, if you have an unbalanced centrifuge rotor, that rotor is going to start spinning and then be thrown off kilter with some fairly catastrophic results. And so you don't want to ever spin an unbalanced centrifuge. So always add tubes such that the rotor is balanced. And so these Eppendorf centrifuges fit your Eppendorf tubes. But then we also have another version of centrifuge that fits your 15 mil Falcon tubes. And so again, you're never going to add a single Falcon tube to these centrifuges. You're always going to balance those and then spin those according to what setting you need to and what setting your lab protocol calls for. And so these are the large pieces of equipment that you'll be using in Bio 120.